In this lecture, we'll discuss about DNA isolation, digestion, and ligation. We have previously discussed gene cloning, and the first step would be to isolate the DNA. In order to do that, we first need to culture the cells that contain our target gene. Then we lyse it with a detergent like SDS. Once we have lysed the cells, we centrifuge it and all the cell debris precipitates and the cell contents are released into the solution. Then we treat it with phenol chloroform. They are organic solvents and they precipitate proteins. So in the next centrifugation step, we can separate proteins from nucleic acids. The remaining nucleic acid solution can be treated with RNase H to eliminate RNA, and then we can precipitate the DNA using absolute ethanol. Once we have our DNA, we can dissolve it in a buffer and we'll have our DNA solution ready. In the next step, we digest our DNA and the vector molecule with a specific restriction enzyme. There are various restriction enzymes which are produced by different bacteria. For example, EcoR1 is produced by the bacteria Escherichia coli. Hind3 is produced by Haemophilus influenzae. Every restriction enzyme has a specific restriction site. For example, the restriction site of EcoR1 is GAATTC, and the restriction enzyme cuts between G and A. The cut of the restriction enzyme is usually uneven and the ends usually do not have base pairs. Such ends are called sticky ends or cohesive ends, and they help in the attachment of the vector to the target gene. So what we do is we add buffer, the DNA and restriction enzyme. Usually we use around 12 to 15 microliter of DNA and 1 microliter of restriction enzyme. Very small quantities. If you remember, 1 ml is 1000 microliters. So we are using only 15 microliter and 1 microliter of restriction enzyme. Similarly, we take another test tube and we add our vector DNA molecule and add the same restriction enzyme. Then we incubate these tubes at around 37 degrees for 16 hours. So in the end of this step, we have our target gene which has been digested by the restriction enzyme, and we also have a vector molecule which has been digested by the same restriction enzyme, and they both usually have complementary sticky ends. The next step would be to ligate, that is attach our target gene with the vector molecule. So we add buffer, vector DNA, target DNA, ligase and PEG, that is polyethylene glycol. Usually we need around 100 nanograms of vector DNA and 300 nanograms of target DNA. And we use only 1 microliter of ligase enzyme. Polyethylene glycol is known to help in the ligation process. Once we add all these to our test tube, we incubate it at 16 degrees overnight. At the end of the ligation step, there are two possibilities. Our target DNA has successfully combined with the vector molecule and we have a recombinant vector. Or the vector molecule has self-ligated and it does not have our target gene. So now, after ligation, our test tube containing the ligation mixture can have either the recombinant vector or the self-ligated vector. We need the recombinant vector for our experiment, but as they are both present in the same test tube, we cannot differentiate one from the other. We can only do that after we complete the next step, that is transformation. We will learn about that in the next lecture.